Hi, everyone. Thanks for joining today. We'll get started on the hour. Hello, everyone. Thank you for joining today and welcome to the NOAA Omics Seminar Series. I'm your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the past few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on our NOAA Omics YouTube, and they're posted on our NOAA Omics website. There should be a link to that website in the chat now. As participants, you are muted, but feel free to type questions in the questions for staff box throughout the presentation. And at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Making NOAA Omics Data Fair, the NOAA Omics Data Management Guide by Dr. Katherine Silliman. Now I'll go ahead and allow screen sharing while Katherine sets up her slides.
All right, can you see that?
Not yet.
There you go.
Oh, there we go. Okay. Dr. Katherine Silliman is the head of the NOAA AOML Omics program in Miami, Florida. Her research focuses on developing omics applications and data pipelines for enhanced marine biodiversity monitoring to support fisheries research and ecosystem assessments. Prior to joining AOML, she worked on developing and applying omics methods for conservation and fisheries management across the southeastern United States. She graduated from the University of Miami in 2013 and then completed her PhD research at the University of Chicago on population and functional genomics of Olympia oysters. Thank you, Katherine, for presenting today, and I will now pass it off to you.
Thanks a lot, Nicole. Yeah, hello everyone. Excited to be talking to everyone today about data management efforts. Maybe not the most exciting topic, but one that hopefully at the end of the talk you'll feel is important for all of our research. And before I really get into it, I want to acknowledge that everything I'm talking about today came out of a lot of hard work and discussions from lots of folks in the NOAA Omics Working Group, and in particular, Luke Thompson, Zach Gold, Sean McAllister, and others of our data and bioinformatics group. So Nicole already gave the quick overview about what we mean when we say omics. I just really want to highlight here that omics data is increasing rapidly across NOAA-funded research. And that is due to integration with existing oceanographic research cruises, the development of autonomous samplers, and also just the falling cost of sequencing can make it increasingly easy to get a lot of data in hand. The challenge then is figuring out what to do with it. Omics methods also vary widely and evolve rapidly, and so this often results in requests for guidance and support to manage this large quantity of data at all levels of users. So another term I want to make sure you're familiar with is this idea of fair data. And this concept came about in order to optimize and support the reuse of data that comes out of research, all kinds of data, not just omics data. And the F stands for findable. And so if you want to reuse data, you have to be able to find it. And to help with that, it's recommended that you assign globally unique and persistent identifiers, such as a DOI that you might get when you publish a manuscript, and also that your data is indexed in a searchable resource. The A is for accessible. And so once your data is published and out there, you want it to be in a format that is openly retrievable and ideally free to access. I is for interoperable, and so this involves the concept of data standards, which are terms and controlled vocabulary that are formal, accessible, shared, and broadly applicable as a way to represent knowledge. And these data standards often come about through community discussions. We also, in order to make your data interoperable with other folks' data, it should be in a machine readable format, in particular in open formats that are not only able to be accessed via proprietary software. So CSV would be a good example of a file format that is machine readable. And then reusable. If you've ever tried to reuse someone else's data, you know how important it is that it is very richly described about how they came about generating that data, and that it includes accurate and relevant attributes. Data needs to have clear and accessible data usage licenses and meet domain relevant community standards. So you're probably asking, why should I care about making my data fair? That sounds like a lot of work. And one answer is that, at least for NOAA funded research and federally funded research for many other agencies, it is a requirement both for intra and extramural funded research. A lot of this stems from a 2013 policy memo from the White House Office of Science and Technology Policy, OSTP, titled Increasing Access to the Results of Federally Funded Scientific Research, where they basically said federally funded research, that data and those results need to be accessible by the public. This has led to a number of clarifications and more clear policy guidance coming out of NOAA, such as PAR, which was the Plan for Increasing Public Access to Research Results, that came out in 2015. And then more recently in 2022, OSTP said, all right, you guys really need to update your public access policies as soon as possible, no later than December of 2025, so that research publications and their supporting data that come from federally funded research are publicly accessible without an embargo, so without a delay, on their free and public release. NOAA's updated response to that is to come out with a policy handbook titled the NOAA Data Management Handbook. That the first version is out now for comment. The second version will be released in October. And this handbook has been and will continue to guide a lot of NOAA's data management requirements and policies going forward. But beyond just caring about fair data because you have to, there's a lot of benefits to individual researchers and their research groups. Making your data fair can lead to more citations because it's more findable. It can be reused in new ways. It leads to more efficient use of research funds and new discoveries through synthesis across research groups and domains. Having reusable standardized data will save you time in the long run. If you've ever tried to collate information and data that was collected by 5 different people in one lab across 10 years, and everyone used a different term for the temperature measurement, you can see how that can save you a lot of time. And also, it's really crucial for generating AI-ready data and supporting future applications that we may not even have thought of yet. And that's all well and good, but in reality, management of omics data is complex, sometimes more so than data that's generated by a buoy or by a satellite. And that's because data from sample collection all the way through processing goes through many different steps, and there's distinct metadata that comes from each of those steps. Now, the term metadata can mean different things to different people, and so here we're often using it talking about the contextual metadata of the data itself. So the what, when, where, and how about that data collection. Metadata also can refer to metadata about the project as a whole. And so that might be more the who did it, why it was done, and importantly, where the data can be found after it is published. And so all of the different types of metadata, from metadata about sampling, processing, analyzing, all of these are really critical for data reuse and being able to integrate data across projects and across space and time. So that in itself adds a lot of complexity to omics data management, but it's additionally complex because you produce often lots of different data products from one study, and those products have to be submitted to different online repositories if you want to make them publicly available. Furthermore, each of those repositories requires different formats and data standards. And so we have begun mapping out the ideal data lifecycle for NOAA Omics data using the NOAA Data Management Handbook as a roadmap. And so here is an example flow chart for one particular omics application, and that is a water sample that was processed for environmental DNA to identify species that are present in the environment. And it really highlights the complexity that's involved with thorough data management, even for this one particular application. So to ensure that our data is reusable and fair, it's important to record contextual data and reproducible protocols for each step of the sample and data processing workflow. Raw sequence data is required to be submitted to the National Sequence Repository, which is NCBI. So many journals won't even publish your papers anymore if raw data is not submitted here. And so that is one area where omics data often is at least made accessible. However, processed data tables, like in this example, that have biodiversity information, the list of species and their unique sequences, that table is often not accepted by NCBI. And so it needs to be aligned to different standards and formats if you want to share it on biodiversity platforms. And then, in addition to all of this omics data that is generated, there are often associated biogeochemical measurements that are either collected by the omics researchers themselves or by collaborators. And that, for NOAA data, is supposed to be submitted to the National Centers for Environmental Information, which is NCEI. And then even if you get everything in the right place, there's a challenge of associating all of these data products together in a fair way. And this challenge of linking the variety of data streams that can occur from a single research cruise or sampling effort is really shared across NOAA-funded projects. And so while we haven't solved this challenge entirely, later in the talk I will highlight how we're starting to address it for omics data. Now that data flow diagram I showed earlier is a typical workflow for environmental DNA data. However, omics methods and the resulting data types are extremely diverse. And so this table highlights the variety of data types, formats, and designated repositories for the omics methods being employed at NOAA. However, it feels like new methods are developed almost daily. And so this range of data types and omics applications kind of restricts our ability to make a single, all-encompassing NOAA omics data management plan. And other distinct challenges for omics data include the large, sometimes terabyte-sized data that itself can be processed into multiple new, large, intermediate, and final data sets, all of which should probably be archived as well. So if I haven't scared you too much, so because of this really diverse types of omic data being generated at NOAA and through our partners, and this still evolving data management policy landscape, we have developed a data management guide in order to help assist users navigate this process. The guide is a living online resource intended to provide best practice guidance and tools for omics data management, including resources to draft your program or project-specific data management plans. This guide also stems from repeated requests for guidance from program managers who are funding omics projects for the first time. And it was really built from the principles that are provided in the NOAA Data Management Directives Handbook. And so I'll have this link multiple times in the talk, but here is the QR code that takes you to the guide. So at the start of the guide, we provide 4 key requirements or recommendations for NOAA omics data in order to meet policy requirements. First, to prepare a data management plan. And to help with this, we provide a NOAA Omics data management plan template. The guide includes text on who should be filling out a template and at what level. Second, to submit omics data, such as DNA sequences, to the appropriate public data repository. In many cases, this will be NCBI. Third, that projects, including environmental measurements, should submit those data and metadata to NCEI or the appropriate repository. And lastly, we strongly recommend cross-linking persistent identifiers such as DOIs and the accession numbers that are generated by NCBI so that all data and metadata from a project can be discoverable together. Now, one of the products in the guide is this NOAA Omics Data Management Plan template, which was adapted from the Data Management Plan template found in the NOAA Data Management Handbook. Now, based on stakeholder feedback on initial drafts of our template, we have collapsed some of the official handbook template headings and then also added prompts to be more concise and omics relevant. So data management plans are already required for a number of NOAA funding programs, and starting in October, there will be increased efforts across NOAA programs to start collecting DMPs and tracking policy adherence. So we have been working closely with the NOAA data community to put together this omics specific template that ideally captures the nuances of omics data management while minimizing the burden on those who fill it out. But beyond just being a requirement, developing a plan using the guide can actually be really helpful for mapping out and improving your data management workflows. The next recommended guidelines involve submitting data to repositories, which almost always requires aligning your data to existing standards. So if you've ever submitted your data to NCBI, you're actually probably familiar with the MIXS data standard, even if you didn't know it was called that. MIXS standing for minimum information about any sequences. And so when you submit to NCBI, there's a Excel spreadsheet template that they give you. It has controlled terms for collection date, for latitude, longitude, geography. All of those are terms coming from this data standard. However, as I mentioned, NCBI doesn't take all the kinds of data products that we want to share. And so another common repository include these biodiversity platforms. And those use a different data standard called Darwin Core, which was developed by the TADWIG consortium. And these have lots of overlap. Some of the terms are exactly the same, but even across these two different standards, we have found that they don't always meet the data management needs of the NOAA Omics community. So in addition, we have been working together in collaboration with a multinational group based in Australia on developing checklist of standard terms for omics data that ideally one day can be included in the MIXS NCBI framework, as well as available to journals for evaluating fair data practices. So we've taken those terms from those different standards, as well as some of the custom terms we've developed, and created Google Sheet templates to help users organize all of the data and study metadata that are generated throughout an omics project lifecycle. So these are built using Google Sheets. If you go here, you'll go directly to the website with all the documentation on using these sheets. That's also linked in the guide. The way they're set up is that distinct templates exist depending on the type of omics method you're using and the environment that the sample derived from. For example, you don't need soil pH if you're working with water data, etc. And so these templates go a long way towards improving interoperability of NOAA Omics data sets, because from the very beginning, we're using controlled vocabulary for contextual information about our data. And also, it's to help streamline the submission of data to repositories. And so These templates are built directly around the NCBI submission templates, and I can show you a bit how that is set up in a minute. I also want to point out that Baden Willems, who's a software engineer at AOML in Miami, has developed a pipeline that will auto-generate these templates for pretty much any environment type, which is really exciting. And so if you go on the website and there's a specific case that you don't see a template for, reach out to us. We'd be happy to help you get that set up. And so just to show you a bit how these are organized, each tab in the Google Sheet is a different type of metadata that you're recording throughout the project lifecycle. The first one is just information about the study itself, the name, the funding source. The second one, the sample data sheets, have information about the individual samples. And so with the example of environmental DNA, each row here might be one discrete water sample that was taken, and the date and the location and the methods used for that collection. We then have prep data sheets, and here each row is a sequencing or omics library preparation done on a single sample. So you might have multiple of these if you did different omic preparations on the same sample. And it contains information like the primers that were used and the DNA extraction kit protocol. It's set up in a way so that it has both the controlled terms that are required for submission to NCBI sequence read archive, as well as some of these more controlled vocabulary terms for methods that NCBI does not track. And yeah, and with the concept that it makes it, at the very least, really easy to just download the spreadsheet and upload it directly into NCBI. And we have guidance on how to do that on the website. Now, as I mentioned a number of times, NCBI does not take a lot of the process data that we also want to make publicly accessible. And so based on current national and international positions, one of the recommendations that we put forward in the guide is that biodiversity observations, such as those that come from short DNA sequences, be submitted to biodiversity platforms like OBIS, which is the Ocean Biodiversity Information System and is funded by UNESCO, or GBIF, which is global, has a number of government funding, through the International GBIF network, and pulls biodiversity data from many other biodiversity repositories such as iNaturalist, eBird, and also OBIS itself. One distinguishing feature is that OBIS is marine only, and OBIS is actually archived on NCEI for long-term preservation. So that can kind of kill two birds with one stone in some cases. So although there is this international guidance on how to submit DNA data to OBIS, I'll admit that the process is complex and user-friendly tools are still in development. But a couple do exist. And so one, to fill this need, we have developed a Python workflow to prepare specifically eDNA data for submission to OBIS. This workflow facilitated the first eDNA data from a NOAA Omics lab going to OBIS. GBIF also has and so one factor I'll say about this pipeline is it's built around Jupyter notebooks. It uses Python code, at least some entry-level background and familiarity with those tools is required to take advantage of this. Another option that has come out very recently is a no-code solution from GBIF Secretariat. It is a prototype, but this is a GUI platform where you upload your data and it does a lot of the data formatting and conversion for you. It's just an example of some of the benefits of making your data available on these platforms. This is a case study data that we submitted last year from the Ocean Acidification GOMEC-4 cruise in the Gulf of Mexico. This cruise used 16S and 18S markers for amplicon sequencing to look at protist, bacteria, and archaea diversity. Since submitting these biological observations to GBIF and OBIS. The data has been featured in over 3,900 downloads and 7 citations. And so it's exciting to see this data getting out there and getting used, again, in ways that it wasn't even originally planned for. So just to spell it out, if you want to easily share your omics biodiversity data to OBIS or GBIF, one, you want to make sure that your data includes spatiotemporal biodiversity observations. In many cases, experimental work that came from lab environments is not necessarily ideal for submission to these biodiversity platforms. But if you have eDNA metabarcoding survey, if you have qPCR surveys, and even field tissue collections that have some associated genetic data, those are all great types of data to be shared on these platforms. You then would want to use a template to help standardize your data and metadata, such as one of the templates that we provide, or you can even start with the Mixes templates that you can download directly from NCBI. You then, if you don't know the taxonomy of your biodiversity observations initially, you want to assign that. And so that may be more relevant to these eDNA metabarcoding methods, where you originally just have a list of sequences. You then need to format your data to the Darwin Core standard. And again, there are a couple tools to help with this. And then finally, contact the node manager for USA, GBIF, and OBIS in order to start the process of submitting that data. And that is Steve Hermel, who sits at USGS. Another plug I want to put in is some great work done by Elijah Hall, who works with NCEI, and that is developing standardized vocabulary keywords for omics projects, omics data, and omics methods. And so these keywords are listed in the NASA GCMD, which stands for Global Change Master Directory. And this directory has existed for over 20 years and is a hierarchical set of controlled vocabularies that help ensure Earth science data, surfaces, and variables are described in a consistent and comprehensive manner and allow for precise searching of metadata. And so we're recommending that whenever you submit metadata about a project, to any of these repositories, if it has a place to add keywords, look up what the correct keyword is for your omic method, for your project, and just use that term, as that can really help facilitate searching across NOAA-funded data sets for those that are using omics methods. Some of the remaining data management challenges include this cross-linking of omics data across repositories. So as I mentioned, our current strategy is really to list all project-specific DOIs in the approximately appropriate metadata field within each individual repository. Unfortunately, not all repositories have designated fields to allow this kind of cross-linking. And so this is something we're working with the NOAA data community to address. In any case, while this may help a knowledgeable data user eventually find all the relevant data for a sample or project, it would be ideal if data linkages could be listed in a centralized location. And another issue that's really shared across NOAA are the inconsistencies among line offices and funding programs in terms of the resources and protocols for making that project metadata and environmental data available on NCEI. And as omics methods are really used across the agency, it can be challenging to provide standardized guidance. Finally, making more accessible tools to streamline data archival practices. We've made a couple early steps into providing templates, providing resources, providing code, but more work can certainly be done to make that an easier task on a typical researcher. And then also we're continuing to develop guidance for all types of omics data generated through NOAA. And so we're actively interested in developing guidance for genomics that come from tissue samples, metabolomics, and qPCR. If this is a space that you have thought a lot about or would like to contribute to, please reach out. And I also want to highlight a couple of other NOAA Omics resources, one being the NOAA Omics Technical Portal. And so this is a new living website repository for NOAA Omics resources. It includes sampling and lab protocols, bioinformatic tools, links to NOAA Omics related code, and training resources. I in particular want to highlight some of the standardized ways folks have been recording their sampling and lab protocols using this BBOP, Better Biomolecular Ocean Practices format. And so yeah, here's what the technical portal looks like currently. It's simple, but it's a place to be collating a lot of these resources into one place. And then these BBOP protocols, they're built using templates. This is one that was put together by Zach Gold's group at PMEL. And it's a way in a controlled, organized method and machine readable format, provide all the information needed to do a protocol. I believe this one is for 12S PCR, but they also have one for collecting from CTDs. So definitely go check those out, and highly recommend if you have a protocol that you would like to be featured. If you want help getting it set up in that format, let us know, as that would be, yeah, we would love to highlight more resources on here. In particular, if you have feedback after checking the portal out, reach out to Zach Gold and Nicole Miller. And if we're missing your NOAA Omics code repository, protocol, or online resource, we have a form that's linked to on the website for suggesting that to be added. And just to wrap up, I want to again point out sort of the four key bits of guidance we have, and that is preparing a data management plan, submitting your omics data to the appropriate public data repository, for example, NCBI. Make sure you're submitting your non-omics environmental data to NCEI or the appropriate repository, and make sure you're cross-linking persistent identifiers so that all data and metadata from a project can be discoverable. And so the guide was really written to serve a variety of end users, both internal NOAA staff, program managers who are supporting omics R&D, and NOAA grant recipients. In particular, we think this guide will help researchers who are incorporating omics data for the first time and are unfamiliar with the unique data repositories like NCBI and metadata expectations that are out there. It came from contributions of lots of members of the NOAA Omics community. It's incorporated feedback from a number of beta testers, but we're always seeking more input from the omics community and the NOAA data community at large. So please check out the guide at the link in the QR code. If you have feedback or ways that it can support your omics data management efforts, you're welcome to reach out to myself. And with that, we can open it up for questions. Thanks for listening.
Katherine, what a wonderful presentation on the hard work that you and the NOAA Omics team have been putting together to develop these tremendous resources, the NOAA Omics Data Management Guide, associated templates, the GCMD verbiage, the NOAA Omics Technical Portal. This has been a really productive year for your team and NOAA Omics. Thank you very much for sharing with everybody these new resources that are out. We'll go ahead and take some questions today from the audience and see where the conversation leads us. We don't have any yet, so feel free to take some time and I will start us off. So these, it's a NOAA Omics Data Management Guide, but can non-NOAA staff use this guide, and how would you recommend them using it?
Yeah, so there's a lot of content in there for just managing omics data in general, in terms of like these concepts of using standards, making your data fair is something that I think everyone should strive for. If you have research that comes out of a NOAA-funded project, these policy requirements do technically apply to those as well. And so even if you're not NOAA staff, many folks do receive NOAA funding. And so this is certainly relevant to those folks. And yeah, but otherwise there might be some specific requirements on there, such as submitting data to NCEI that would maybe not be relevant to a non-NOAA funded group, but I still think there's a lot there that can be helpful. And the template certainly could be used by anyone working with that kind of data.
Thanks, Katherine. I will start us off with another question. You mentioned that the data type right now can be tailored to, the guide can be tailored towards eDNA, but you and your team are working to integrating all types of genetic data into this guide. Is that ready or coming in the near future?
Yeah, so the guide has a lot of text for lots of different kinds of omics data. The resources that so far have been mostly eDNA focused include those study data templates. Right now, the existing ones are for environmental DNA samples taken from water, sediment, and host associated. Yeah, the tools and guidance for getting your biodiversity observation data on OBIS and GBIF, those also are generally eDNA focused. But the guidance has plenty of text on there for working with tissue-based omics methods. It has a little bit of guidance for metabolomics, for qPCR, but yeah, there can certainly be more work done to expand that.
Definitely, and we are very much looking forward to it. Okay, our next question is from Carol, who asks, what are the coming greatest challenges and improvements to be made, in your opinion, thus far? To I think the field, but also maybe the guide.
I think specifically for, yeah, I think one of the biggest challenges is integrating environmental data that is shared in one repository with sequence data that's shared in another, and making it so that researchers can easily find those, but also ideally so that the AI crawlers that are certainly on their way can even be able to link all of that information together. And so I think that that's sort of the next big challenge that we're working on.
Thanks, Katherine. Our next question is from Zachary, who says, wonderful talk, Katherine. What do you think is needed to mobilize omics data into these global repositories? If you were queen for the day, what would you do or need to support these efforts?
Man, if I was queen for a day. No, I think, so yeah, a number, yeah, I was attending the National Marine eDNA workshop about a month ago, and thinking about the potential large scale of environmental data, biodiversity observation data that could be generated in the near future, asking individual PIs to be doing this kind of data formatting, even if we have relatively easy, straightforward tools, seems like a major challenge and workload. And so in the interim, that's the recommendation, but I think in the hopefully not too long term, we have more automated processes for helping folks get their data formatted and getting it submitted automatically. So we've talked about maybe centralized databases, centralized data processing centers where you can submit your data in a more relaxed framework, and it does a lot of this conversion for you. So I think if I were queen for a day, I would fund a bunch of data managers so the rest of us can get back to doing science, but also fund some more centralized processing of submitting data to these repositories.
I don't think I could agree more, Katherine. Great response to that one. You have my vote. All right, our next is from Frank, who says, thank you, Katherine, for a wonderful presentation. I was wondering what internal discussions there may be on common guidelines on harmonizing eDNA/omics methods, basically to ensure that things are comparable from one place to another over time. Thanks again.
Yeah, I think there are, I'm aware of a number of conversations about, standardizing methods. I think those are important to have, but I also think that new technology, new approaches come out all the time. And so realistically, we probably won't say everyone process your data this exact way, because next week a better way might come out. What I think we can and we should do is very clear, standardized metadata, recording metadata about how samples were processed, how samples were analyzed, and how that data was curated. Because even if it's not all done exactly the same, if you at least know exactly how it was done, then you can start to incorporate statistical tests, you can account for those variations in methods. And so it may feel like a lot of work to very clearly detail all of the methods that went into generating data, but I think that's critical if we ever want to do these kind of large global synthesis research.
Thanks, Katherine. Thank you. It looks like we have room for one or two more questions. This next one comes from Nicole, who states or asks, thinking back to the flow chart showed in the beginning with eDNA as an example, field and lab protocols should be adequately described in any publication. So how can this NOAA Omics data management guide enhance the fairness of data outside of a publication?
So what I would say should is, they should be, but that information being stored inside of a PDF is not machine readable. It's not easy to access. And so, if we start thinking beyond, I just want to reuse the data in your single paper to, I want to reuse data in 100 papers or 1,000 papers, that's where saving your methods in a protocol that itself has a DOI using a standardized machine readable format. That's where that can be really critical and really helpful. It also, you might think that you put every little bit of how you did something in a paper, but using templates and using controlled vocabulary can actually help you realize like, oh, I did not actually say all of the temperatures of my PCR protocol. Things like that, having, I think, standardized ways of sharing that beyond just free form of the paper can be really helpful.
Thanks, Katherine. Thank you for that. With that, it looks like we've come to an end with our questions. I really would like to thank Katherine for joining today and the great presentation on the NOAA Omics Data Management Guide and making NOAA Omics data fair. And thank you all for joining us today and your great questions. We are very lucky and happy to have you join almost nearly 75 of us today, which is a huge feat for the NOAA Omics program. Our next seminar will be on August 21st at 2 p.m. by Nicole Housley, who will present on life lessons from a new eDNA laboratory. So a great follow-up to this month's seminar Thank you very much, everybody, for joining, and we'll see you next time. Any final comments, Katherine?
Nope. Make your data available. That's all.
Thanks, everybody, for joining today.
Thanks, Nicole.